I think you're gonna have to use the keyboard.
Okay, keyboard. Okay, I think it's working. I wasn't nervous at all, so. Wow, so first I would like to say something about myself. So just to introduce myself, I'm Brazilian. I studied biological sciences in the Universidade Federal de São Carlos in Brazil, and I also did my PhD there in ecology. And currently I'm working at the Universidad Nacional Autónoma de Mexico in Mexico City. I am half Japanese and half Brazilian. So this is why I have this strange name. Well, so I divided this presentation in three main topics. One will be just an introduction about parasitoids. I believe most of you already know lots of things about parasitoids, but I decided to give this introduction because some people work with different things. So, and I think it's more interesting to talk about them than talk about myself, right? And also I will talk a little bit about the taxonomic gap and the taxonomy impediment that are huge things in taxonomy right now. They always been. So I think it's important because this is where I think the most of the efforts have to be done in taxonomy. And I'll talk something about my current projects as well. Okay. So let's start with the parasitoids. I think most of you already know, but parasitoids are mostly insects that develop on other insects most of the times. I like to think of them about like they would be viviparous like us, but instead of using a mom's womb, they use another organism, and they are born as adults instead of babies. And they kill their mommies in this. So one thing that's really interesting about parasitoids is that in the case of hymenopterans, which are the great majority of the parasitoids, is that they have these venom glands and this, which is part of their reproductive system, and these ovipositors that are, as you can see, really close to the ground plane of the holometabolous insects. The pteroid actually. So they use these ovipositors to reach hosts in very different places, even in very deep burrowed in woods, for example. And the venoms are really important because they can paralyze their hosts, and sometimes permanently, sometimes just for a while. And these are really important pre-adaptations for the success in the parasitic way of life. Another aspect of parasites that's really interesting for me is that they can sometimes manipulate the host behavior. In this example, which I'm showing to you, this is a caterpillar that was parasitized. The parasitoid larvae just left the caterpillar and spun their cocoon there. And the larvae is like almost dead, but still alive and protecting the cocoons of the parasitoid. So this is something that they can do. It's really interesting. There are several examples of that. And I think it's something related with the zombie anti-fungus that is something that it became popular in popular culture because of this TV series, right? So what these adaptations do is to improve their success in reproduction and so on. Another thing that's really important, I think many people study parasitoids because of this, is their capacity of regulating other populations, right? And I think it's more important than reducing populations is the fact that they can maintain and persist in long time in a dynamic equilibrium. So this is something that we observe in some. How can I say it, like in experiments in nature, when you introduce a parasitoid species in a different place, and you can see the same trends that you see in these consumer resource dynamic models. So of course this is really interesting in terms of ecology, but it's also interesting in terms of economy. So these ecosystem services have economic value, and sometimes you can actually put currency value on those services, but actually the food security that they can provide is really what most of us are like concerned about, right? So just to give some examples of how many billions of dollars can worth these services. And other thing that's really interesting about parasites is that they comprise like a huge amount of the terrestrial biodiversity. If you think about insects that have more than half of the species in the terrestrial habitats, and you put the parasitoids on it, there would be like at least 20% of the insects are parasitoids. These are old estimations, and we'll talk about newer estimations later, but just the fact that they are like this huge part of the biodiversity, it's something that it's worth studying, right? And of course, they're beautiful and they're really diverse. Some of them are really tiny, as you can see, compared with the tip of the pencil. And so I think there are many reasons to study them, right? Like they're very cool biologies and their diversity and the capacity of manipulating their hosts. So, but actually the thing that made me start studying them was that I had a friend that needed help in her grad studies. So I went for field work and sample sorting, and I became to know the parasitoids because we actually don't know them in the undergrad studies, right? So unfortunately, this is not a not noble reason at all, and I'll show in the future why in the next slides why I think it's important to start this way, right? Anyway, let's talk about Braconids. Braconids is the group that I focus most of my research. They are the second largest family of hymenopterans. They are only behind the ichneumonids, which, for some coincidence or not, is the sister group of the Braconids. They form the superfamily Ichneumonidae. And these are the two largest families of Hymenoptera. Just to give you an idea, the Braconids have more than 1,000 genera. Okay, so it's a lot of diversity. They are spread worldwide. The phylogeny of the group is really interesting as well because they seem to be one of the most basal groups of apocryphons. Apocryphons are the hymenopteras that have this wasp waist. Okay, so most of the diversity of the hymenoptera is in the apocryphons, including the social insects, the ants, the bees, and the wasps. So they have this long history of evolution, and they have lots. They have most of the species many of the species are in this group. So they're really successful. We can say they're really successful in diversifying. So I think this makes them even more interesting than they are than the other groups. To recognize them is quite easy. I don't know if I can point with this. Is this a pointer in this? What is
You can use the mouse.
the mouse? Okay, thank you. Okay, so one character that you can always check is the presence or absence of this cell. This is called the coastal cell. In all the Braconids and Ichneumonids, it's absent. So all the two coastal veins are fused, and the filiform and have a relatively long antennae. So this is how you can easily identify one Ichneumonidae. And to differentiate the Braconids from Ichneumonids is easier as well. You just need to check the second and third turgets of the metasoma. If they're fused, it's a Braconid. Okay, it's pretty easy. So if you find some, you can bring it to me. Biology of Braconids is also really kind of straightforward. Most of them are larval parasitoids. It means they put their eggs in the larvae, and not the adults, but before pupating, they leave the host still in the larval phase most of the times. Okay, there are some evolutionary trends of positivity in earlier instars, including in eggs, and some braconids will emerge from the pupae, especially the ones from fruit flies. Okay, so most of the hosts are the holometabolous insects, the Lepidoptera, Coleoptera, and Diptera. There are exceptions. Some of the most important exceptions are the parasites of aphids, which are important for biological control, of course. Some groups are parasites of adults, which is really rare in the parasitoid world, even outside the braconids and echinomonids. And some are phytophagous, which is not so rare in the parasitoid world. And one really, really interesting thing about the parasitoids is in the braconids and echinomonids, they have this domestication of virus. So some groups of braconids and some groups of echinomonids have this viral genomes integrated into the wasp's genome, and the virus lost the capacity of reproducing by themselves, but they can still produce virulence proteins, and these proteins are likely to help to aid the parasitoid to suppress the immune system of the hosts. So this was first discovered in these groups, and we now know that there are many other groups that have this kind of domestication. And most of this domestication happen in groups that are parasitoids of Lepidoptera. Okay, with one exception here is the parasitoid of Diptera here. Well, so this was the fast introduction I had about the parasitoids. Now I would like to say a little bit more about the taxonomic gap and taxonomic impediment. I guess most of you know about it, so I don't need to get into much details of it. I just need to say that the taxonomic gap is really huge, and we have the biodiversity crisis, and this makes the necessity of studying the taxonomy of many groups much more important. And we have all these dark taxa, including the parasitoids, that needs more efforts than ever. And then in the other side, we have the taxonomic impediment. So taxonomies are like we are being extinct, right? And the proof of it is that I'm here, right? Because yeah, you understand. So I want to do more details on how do we quantify the gap and how do we deal with this problem. So first, quantifying the gap. So species diversity. We can think about species diversity in terms of niche availability, differentiation. So and when we think about parasitoids, almost the entire niche of the parasitoid is their hosts, right? So one way of think about it is if parasitoids are the number of species of hosts that there are for parasitoids, you would know the number of parasitoids there are. So we have 1 million species of insects. If every insect has one parasitoid, we would have 1 million parasitoids, right? Of course, it's not as simple, but having this in mind, there was a publication in 2018 that the conclusion of the publication is that the hymenopterans, because of the parasitoids, are much more diverse than the coleoptera. And the coleopterists doesn't like it, right? Okay, we don't mind. So what does it mean? The estimations that this work did were based on only 4 genera, okay, of the major groups. But the conclusions were really interesting because they saw that at least close to 1-to-1, which means one parasitoid species for one host species. This would mean that we have 1 million species of parasitoids, right? This is huge, and this is the lowest estimation. But it seems a little unrealistic if you think about, I will just extrapolate from 4 genera. Okay, so I wanted to take this to present some of my results from my PhD, in which I studied tritrophic quantitative food web based on 2 species of plants in a tropical region in Brazil. So in my results, I found that one species was really a dominant species, but these only species had 21 species of parasitoids. Okay, and this is interesting because the parasitoids are divided into 6 guilds, and they can share the same resource by segregating the niche into, especially in time, but also in space. But the other part of this is that, in general, the results of the parasitoid host ratio was almost the same as the one that found in the 4 general thing, right? So I have this 1.15, which is almost is between the range that they found. So I think it's kind of realistic to think that we have at least 1 million species of parasitoids to describe, which is a lot. If you think we have in Hymenoptera, we have kind of about 90,000 species described. So it would be 10% or less of the existing species. So it's a huge gap. Okay, and I put this example of one in Brazil of one subfamily that I was focusing in. And in this subfamily, in particular in Brazil, we have at most 2% of the species described. So lots of work to do. And I'm saying all of this because I think this is the task taxonomists have, and we cannot just do other stuff because it's more fancy, right? So we have to deal with this all the time. Another example is doing surveys in local surveys for 1 year. In one local survey from 1 year, you can find much more species than they were described in the entire history of taxonomy. So it's common. You go to one place, you collect for 1 year, and you have more species than you have described for the 200 years of taxonomy. Okay, so this is some examples of works we did in Brazil. So how do we deal with this problem? So actually, I don't know. Well, one of the things I think it's important is to train and educate new taxonomists, right? But it's just solves half of the problem because they have to continue their work in the future. So I was thinking about it, and I draw this network of people that were all, they are all PhD doctors that are studying taxonomy of parasitoids right now, and they are all related with Angélica Penteado Dias, which is one of the first persons to start studying parasitoid wasps in Brazil. So the major thing I want to show you is that if you see this, you say, okay, in Brazil, there are plenty of taxonomies, right? This is the first impression, but if you see it more closely, you see that the lowest first people are the first generation. And even the first generation that were, they get their PhD 30 years ago, they couldn't get much of their students into permanent positions. So what I'm trying to say is even having all those people being trained, even in Brazil or in other countries, you don't have places to put them because of the lack of funding, all of that. Okay, so, and the other thing is, of course, to describe the species, right? So train the students and describe the species, and I have some examples of, and most of the times when you are able to revise a genus in Brazil or in the North Tropical region, you can double the number of species at least. Okay, so it's something that I wish I can do during my whole life. But in sometimes you just can't revise a genus or a group of species because of something that's really common, especially in the tropical regions, which is So in this case, I was studying this genus. I think it was the second genus I started studying. I have dozens of new species to describe, and there was just one species in Brazil. All those species that I found, I never described them. And why did that? Because I'm lazy? No, it's not because I'm lazy, but because I couldn't compare the species I had with the one that I described. And why does it happen so often in the tropical regions? Because there are so many old descriptions that are called superficial descriptions, and Rudolf Meyer calls this the superficial impediment. And it's really something that slows down the taxonomy. And in tropical regions, this is worse because, many people went to the tropical regions, took the specimens to Europe, and this happened for a long time in 200 years ago, 100 years ago. And we cannot use the descriptions to compare with our species and describe them. And we don't have access to the types to know if it's a new species or not. And at that time I was I was totally inexperienced, so I just didn't publish anything about that. But this is really something that changed my way of thinking about things, and this is why I think it's so important to improve the information access. And this would be by taking pictures of the types or describing the types again, doing 3D models or taking the barcodes of them, and all of these things that we can do right now. And it would be really important to speed up the description of species, right? So examples of how we can really speed up the taxonomic descriptions are, especially from the last decade. I don't know if it's not fashion anymore to do this, but it was a time when people published papers with 200 species, combining barcodes with morphology. So I think this is something that we can still do, especially if we do the part of taking the barcodes of the museum specimens to compare. And the other thing that's really important, it's the integration with other kinds of types of projects. Taxonomists collaborating with research in other fields. I don't think I need to say much about it here, because I think here is a good example of how this is done. So I will much more learn than say something about it. So I just want to say that there are some things that in particular for parasitoids that are interesting to say. One of them is that some people tend to think that in agro-ecosystems especially, we know all the species. But at least in Brazil, my experience is that even for pests, sometimes you don't know the species, and for parasitoids, you never know the species. So this is just two examples of this. Two relatively important pests in Brazil, which we try to find the parasitoid species. And I have the specimens with me, but I couldn't even describe them because of the superficial impediment, but they are not known at all. So, and they are doing their job of controlling the pests, right? But we just don't know their names. Sometimes we can do the revisions when, it's interesting because there are two different cases. In this case, in the Apanteris case, there is a revision, so I could describe the species. And in the second case, there are no species at all described for Brazil, so there were no impediment for me to describe that. Okay. And the other thing about the integration of biological control is that sometimes you have to think about the species you have to study complexes, species complexes, and sometimes they're cryptic species. And for that, it's really important to have the integration of different kinds of data, especially the molecular, morphological, and biological data. So this is something that I've been doing with some students for a while. And the other thing is to study the parasitoids below the species level in the gamma taxonomy, or the population level, or strains. And this is one classical example of different strains of parasitoids of the same species that were introduced in this Australian region to control different pests, because different strains have different traits. And this is really important biological control, especially if you're doing these introductions. So the classical biological control, because one strain may be harmful for native species and the other not. One strain may control one species, but not the other. So, and this is a really interesting case because they have been studying this for a long time, and there are lots of information about hybridization and so on. So I'll stop here and talk about my groups. How are we? Good. So now finishing my second part, starting the last part, which is about my current projects. Currently, since last year, we started studying the one subfamily of Broconids called Rogadines. Rogadine is a really interesting group of parasitoids because they do something that no other parasitoid do, which is they mummify their hosts. So all of them are parasitoids of Lepidoptera. So they parasite the Lepidoptera caterpillar like other parasitoids, but in the end of the development, instead of leaving the body of the caterpillar, they stimulate the skin of the caterpillar to harden. So the skin of the caterpillar becomes the cocoon, where they pupate and then they leave the caterpillar. So this is a synapomorphy of the group. So it's easy to recognize them if you know the mummies. The adults are not so easy to recognize. But anyway, so one of the first objectives we had in this project was to use the genomic tools to study the phylogenetic of one genus, which is a huge genus with about 700 species described right now. This genus is distributed in the entire world, and there were like several attempts to divide this genus in different subgroups, but there's a problem in it because some of the groups were proposed by people from the old world, and some of the groups were proposed from people from the new world. So these two systems doesn't talk with each other so well. So one of the things we want to do is to propose a global subdivision in subgeneric level. Okay, so for that we are using especially UCEs, which is the ultra-conserved elements of the genome, which is something that many people are using right now for doing phylogenies. And also we are trying to study the ancestral states of the host associations, okay? So we have some preliminary results, and we can see that we found like several species groups that are really neat groups, monophyletic groups, that you can identify morphologically, and you can see that they are related with one family of Lepidoptera. So this is really interesting. This is only preliminary. We want to do it for all the three. But I think the most important thing of this is to have these divisions of the genus. And why I think this is so important, because it's a huge genus. And the big genus are the genera where the most of the diversity is, right? And people tend to run away of those genera, right? Because it's difficult to revise them. But if you have small boxes of groups that are monophyletic and are easily to diagnose, you can start revising each group. And some groups are restricted for one region of the world, so it's easier. So this is why I think it's important, because we can tackle the taxonomic gap in different ways. Okay. And some side results of this was we were able to clarify the relationships of the tribes in this subfamily already. So this is something that is almost ready for publication. And also we find an entire new tribe. So I think it's amazing to find an entire new tribe. So we did it because, and the other thing is, some of those specimens, I had them from my master's. So I won't say 20 years, but something like that. For many years, I had those specimens. I didn't know which, even the subfamily. I knew it was a Procanium, not only that. And we did the sequencing, and we put in the tree, and we found this. There are 3 new genera, and they form a whole new tribe inside the Rogodines. So just for that, we know the biology. We know they are parasitoids of Lepidopteran. They do the mummies and everything. We just don't know which host they have, but because we don't have any biological information yet. But well, and I think this is a good example of how little we know, right? If you know, you're not just describing species, but whole new tribes and genera. And this is what it's not uncommon to find this in the tropical regions. And the second unfolding of this, one interesting unfolding, is a project that we just started. So we don't have any results yet, but we started to, after those 2 students from Mexico, Alejandra and Diana, they found evidences of those virus in Rogodines, which I don't remember, I'm not sure if I told you before, but there are only 2 events where domestication of virus were found in Braconids, and none of them were in Rogodines. So after that, we decided to write a project to explore it more and to see what is the relation between the endoparasitism and the domestication of virus. So as you can see in this tree right here, this clade is mostly of ectoparasitoid wasps, but this group here, the Rogodines, have this transition to endoparasitism. Okay, and we found evidence of some viral elements domesticated in this group. So, and why it's interesting to see this, because this study here from, I think it's from the last year, they found that endoparasitism and domestication of virus are correlated. So this is why we're trying to think to study this relation and how one thing may affect the other. And the second part of the same project is about something that I'm not really familiar with, but we are trying to explore the presence of toxin-resistant genes in the genome of the parasitoids. And why we thought about it, because there was this recent paper from last year as well, where they found the same mutations for toxin-resistant genes in the herbivores and in their predators. So what does it mean? The predators are evolving the same mutations or parallel mutations to be able to detoxify the hosts that have resistance or not detoxify, but not be affected by the toxins that were ingested by the herbivore. And our hypothesis is that the same thing is happening with parasitoids, because they also have to cope with the toxins that the herbivores ingest. So we will explore this. It's an exploring thing, but we think it could be interesting in this clade here, the Aleogene, because the Aleogene is a group that parasitize Noctuoidea, which, as you can see here, have all the mutations for the GABA receptor, which confers resistance to several toxins. Okay. And also this is interesting in terms of IPM in the context of IPM, because resistance to toxins it's a pre-adaptation for resistance to pesticides, right? And we see often in the nature that parasitoids are also resistant to pesticides as well as the as their hosts. So it could be something interesting to investigate because sometimes you're concerned about how biological control and chemical control can be used together. Right? And in the case of endogenous viruses, it's a little bit more complicated. I tried to explain, I'm not sure if I can, but one thing that is observed in virology is that hosts that are infected with one virus, sometimes they get resistance to other infections because I actually don't know the physiology of it, but it's an interesting thing to ask for the future, like because it's the use of virus for controlling, especially caterpillars, is growing a lot, right? And it could be there could be negative interactions between parasitoids and viral control. So it's something that I think it's interesting to think about. And pretty much this is what I had to talk. So I think it's just as a summary to say that integrating genomic tools and barcoding to study the taxonomy of parasitoid wasps is interesting because we can expand, we can take the microevolutionary work and expand to the macroevolutionary work. And so we can delimit species, delimit subspecies, and we can also do phylogenies of the entire family order, okay? And we can also generate barcodes, which is really essential nowadays for doing taxonomy. So all of these things are really interesting. Now, this is something that I read like 2 weeks ago, this paper, that they could use genomic tools to differentiate the host and the parasitoid gene. So you don't need to rear the specimens anymore to know the exact associations between host and parasitoid. So all of these things are interesting for the study of parasitoids, right? And this is it. This is all I had. Thank you very much.